Welcome to another lecture by MedicoMedics, Learning Made Easy, Microbiology and Infectious Diseases, Chapter 3, Microbial Genetics. So in this somewhat lengthy lecture, we will present microbial genetics, discuss bacterial chromosomes, plasmids, DNA structure, DNA replication in bacteria, concepts like transcription, translation, genetic variation, horizontal gene transfer, transposons, and end with a summary. Microbial genetics is the study of how microorganisms inherit and express genetic information. Why is it important? Because it lays the basis of pathogenicity and antibiotic resistance. It is key to genetic engineering and biotechnology. Types of genetic material in microorganisms include chromosomal DNA. This is core genetic material. And extrachromosomal DNA, like plasmids and transposons. Now let's begin with the bacterial chromosome. It is a single, circular, double-stranded DNA molecule, differing from the linear chromosomes found in eukaryotic cells. And it is located in the nucleoid. So that's a region within the bacterial cell that is not enclosed by a membrane. Ergo, it is not membrane bound. And unlike eukaryotic cells, bacteria do not have a nucleus. And that's high yield. So the bacterial chromosome is found in the nucleoid, a region within the bacterial cell that does not have, it's not enclosed by a membrane. And unlike the eukaryotic cell, bacteria do not have a nucleus. Now, since the chromosome is actually quite large, relative to the bacterial cell itself, it is very tightly packed. And supercoiling is the primary method of this compaction, ensuring that the chromosome fits within this small cell. And this supercoiling is maintained by topoisomerases. Let's go with that. And these are specialized enzymes which regulate supercoiling by winding and unwinding the DNA as needed for replication and gene expression. Now, what are plasmids? These are small, circular, extrachromosomal DNA molecules, and these exist independently of the bacterial chromosome. And these plasmids often carry genes that provide bacteria with selective advantage. For example, or including antibiotic resistance, like our plasmids. These encode resistance genes against multiple antibiotics. They also have virulence factors that enhance bacterial pathogenicity. For example, toxins produced in E. coli. And then we have different types. So we have F plasmids, that stands for fertility plasmids. And these enable bacterial conjugation, allowing direct transfer of genetic material between bacteria via something called a sex pilus. We also have R plasmids. These are resistance plasmids, and these carry genes that provide resistance to antibiotics, making infections more difficult to treat. Examples include, for example, plasmid-mediated beta-lactamase production. This is where bacteria acquire enzymes that degrade beta-lactam antibiotics And beta-lactam antibiotics are, for example, penicillin or cephalosporins. So this then leads to drug resistance. Now, why is this relevant to you as a medical student or someone interested in the subject? Well, understanding plasmids is crucial in clinical microbiology and infectious disease management because they play a significant role in antibiotic resistance and bacterial pathogenesis, and of course, therefore, in our treatment strategies. Now let's go deeper and discuss DNA structure. So DNA is a double helix structure composed of two complementary strands. And we have the base pairings which have very specific rules, where A is paired with T and G with C. And A stands for adenine, and it's paired with T, that stands for thymine. You have G for guanine and C for cytosine. Now, these strands are anti-parallel, meaning they run in opposite directions. So one strand is oriented 5 to 3. 5 to 3. While the complementary strand runs from 3 to 5. Now, let's see the differences between bacterial DNA and eukaryotic DNA. So let's first look at structure. Now, bacterial DNA is typically circular, while eukaryotic DNA is linear and organized into chromosomes. Furthermore, bacterial genes generally lack introns. These are non-coding sequences, introns, whereas eukaryotic genes contain introns and exons. We have more detailed discussions of these in our cell biology courses, but for now, just pick up on that there are some key differences. So there are no introns in bacterial genes, while for humans, we will find introns and exons. Furthermore, eukaryotic is linear, right? The DNA is typically linear, while in bacteria, it is typically circular. And what about packaging? Well, bacterial DNA, as we just mentioned in our previous slide, is supercoiled within the nucleoid, while eukaryotic DNA is wrapped around histones and enclosed in a nucleus. That was one high-yield difference. Bacteria does not have a nucleus. Also, its chromosome is not membrane-bound. All of these are high-yield things for you to pick up on. Now then, let's take a look at DNA replication in bacteria. So we have semi-conservative replication, whereby each daughter DNA molecule or each daughter strand consists of one parental strand. So it receives one parental and one newly synthesized strand. This ensures genetic continuity. Now, steps involved here are initiation. This begins at a specific origin of replication and is a unique sequence where replication machinery assembles. Then we have the enzyme helicase, which unwinds the double helix, creating a replication fork. Then we have something called single-strand binding proteins that stabilize this unwound DNA. After initiation, we have elongation, whereby DNA polymerase synthesizes new strands. I think, if my memory serves me right, it's DNA polymerase 3 that is involved in synthesizing new DNA strands in the 5 to 3 direction. And it uses the parental strand as a template. Now we also have the enzyme primase, which synthesizes RNA primers to initiate replication. So after that, we move to termination, and this is where replication ends at the terminus. So replication continues until it reaches this site, the terminus site. And here we have something called TUS proteins that help halt the replication machinery. Now, why is all this clinically relevant? Well, for example, in the development of antibiotics, so some antibacterial drugs inhibit bacterial DNA replication. For example, quinolones. Quinolones like ciprofloxacin target DNA gyrase, which is a topoisomerase, preventing supercoiling and leading to bacterial death. Because here, during termination, the two daughter DNA molecules are separated by topoisomerase 4. Yeah, topoisomerase 4. And this ensures for proper segregation. However, knowing the clinical relevance here is that these medicines like ciprofloxacin targets this DNA gyrase, the topoisomerase, and inhibits it. And this leads to the prevention of supercoiling and therefore bacterial death. So understanding bacterial replication is critical in antibiotic resistance mechanisms and in the development of new antimicrobial therapies. We could also mention rifamycins like rifampin, another antibiotic which inhibits RNA polymerase. So it blocks bacterial transcription. So all in all, knowing these different steps and what happens during these different steps, we can develop antibiotics to stop it. Similar to using antivirals in virus, for example, where we can, by understanding the cycle, the viral replication cycle, we can intervene to block at different stages. And it's the same here. Now, we mentioned in the previous slide rifampin, and we said that it blocks or inhibits RNA polymerase. So why is it clinically relevant? Let's connect the dots. So transcription is the process of converting DNA into RNA. So it's the first step in gene expression where we are converting genetic information in our DNA into functional molecules. And the steps you may recognize from the previous slide, we have initiation. This is where RNA polymerase binds to the promoter region of a gene. This leads to the DNA unwinding, exposing the template strand. Then we have elongation, where RNA polymerase reads the DNA template in the 3 to 5 direction and synthesizes a complementary mRNA strand in the 5 to 3 direction. So RNA polymerase reads the DNA in 3 to 5. And then it has synthesized a complementary mRNA, right? So RNA polymerase synthesizes mRNA complementary in the 5 to 3 direction. And the RNA sequence follows the base pairing rules that we mentioned before, where adenine pairs with uracil instead of thymine, because we're talking about the RNA sequence now. When we talked about DNA, we said that adenine was paired with thymine. All right? But now it's adenine pairing with uracil. And cytosine pairs with guanine. Now, we reach the termination phase. So transcription stops at the termination signal, or at a termination signal. And the newly formed mRNA detaches from the DNA. Now, some key features we can mention to differentiate between bacterial and eukaryotic transcription are the following. So in bacteria, this is occurring in the cytoplasm. Okay? And mRNA is immediately translated. But in eukaryotic cells, transcription is occurring in the nucleus. And mRNA undergoes processing before translation. These processes include, for example, splicing and capping. So again, we repeat the difference between the bacteria and eukaryotic cells, between prokaryotes and eukaryotes. As we mentioned early in the lecture, it wasn't membrane bound, if you remember. It didn't have a nucleus. The bacteria cell doesn't have a nucleus compared to the eukaryotic cell. And so here we also see differences during the process of transcription. Here it happens in the cytoplasm. While in eukaryotic cells, transcription occurs in the nucleus, and bacteria doesn't have a nucleus. Hope it all made sense. Now let's talk about the next step, translation, following from transcription. So translation is the process of converting messenger RNA into a functional protein. And this occurs in the ribosome, where amino acids are assembled in a specific order dictated by the mRNA sequence. And the ribosome 70S, or S, if you remember from previous lectures in this series, stood for Swedberg unit. So 70S in bacteria, remember we're talking about bacterial cells now. If it's eukaryotic cells, we would say 80S. Okay? So in bacteria, ribosome 70S facilitate translation by reading mRNA codons. And tRNA, which stands for transfer RNA, carries specific amino acids, carries or delivers specific amino acids that correspond to these codons in the mRNA. And the genetic code is universal, with each codon specifying a particular amino acid. Now let's look at the steps. So again, we have initiation, elongation, and termination. So during initiation, the ribosome assembles in the start codon, which codes for methionine in bacteria. The small ribosomal subunit binds to mRNA, followed by the large ribosomal subunit. And the subunits are in bacteria, if you remember, the small one is 40S, the large one is 60S. Did you fall for that? Because that's not correct. It's actually 30S, small, and 50S, large one. And this is the ribosome 70S. It's in eukaryotic cells where we have 40 and 60, and we call it 80S. Now, don't get caught up in the mathematics here. The sum of this is not this, but let's leave that for now. That's one of those medical mysteries that we just let slide by us. Now, let's go back. So, we talked about initiation. Now, during elongation, what happens is that the ribosomes move along the RNA, the mRNA, reading codons sequentially. Now, transfer RNA molecules deliver these amino acids, which are joined by peptide bonds. So these amino acids are bound by peptide bonds, or joined actually by peptide bonds, forming a growing polypeptide chain. And the ribosome has 3 sites for transfer RNA binding. It has something called A site, which stands for amino acyl site. And we have the P site, which is the peptidyl site. And then we have the E site, the exit site. And essentially in the A site, the new transfer RNA carrying an amino acid, right? Here you can see in the illustration, the transfer RNA carries this amino acid and brings it in. And in the P site, so we can call this A here, and then in the P site, the peptide bonds form between amino acids. And in the exit site, the empty transfer RNA, remember, it has delivered something, now it's empty. It leaves then the ribosome. Then we arrive at the termination. This is when a stop codon is reached. Translation then stops. A release factor binds, causing the polypeptide chain to detach from the ribosome. So when this is completed, now it detaches. And the ribosome then disassembles, and the newly synthesized protein folds, this one, folds into its functional shape. Now, as always, let's put it in some clinical context. So examples would be tetracyclines inhibit bacterial translation by targeting ribosomes. But how? Well, it targets ribosome 70S. And it prevents protein synthesis from occurring. So this makes then translation a key target for antibiotic therapy against bacterial infections. And we have specific antibiotics targeting 30S, 50S, etc. Now, even though this is not a lecture on antibiotics specifically, for those who are interested, let's go a bit further and specify a bit more so that you understand why you are learning details like subunits for ribosomes. Why are we differentiating between 30S and 50S, for example? Now, let me put this in some clinical context. So we talk about antibiotics. And I mentioned briefly just now that there are antibiotics targeting 30S and there are those targeting 50S. So in the big picture, these antibiotics have something in common, which is inhibiting translation at early stages. And it's essentially interfering with mRNA decoding or with the ribosomal function in general. So we have a group like tetracyclines, and the antibiotics typically found here are doxycycline, for example, and this prevents transfer RNA binding to the A site of the ribosome, blocking protein elongation. You remember when we said A site, and then we had the P site and the E site. I used to memorize this as APE. Don't ask me why, but it worked. Now, once you understand that, oh, there is an A site, right, where it's bringing the protein for delivery, and then we have the binding site, etc., and then the empty transfer RNA leaving. So by understanding at this level, we get that tetracyclines are actually preventing the tRNA binding to this A site, blocking elongation. So we can't get peptide bonds forming between these amino acids. Ergo, we will not be producing these proteins. So we say that these tetracyclines are bacteriostatic. They inhibit bacterial growth without killing them. We also have another group targeting the 30S subunit, which is aminoglycosides. Some of these are gentamicin or streptomycin, etc. These bind to the 30S subunit, causing misreading of mRNA, leading to the production of non-functional proteins. And these are bactericidal, so these kill the bacteria. And now, just to make it complete, we also said we had 50S subunit. So an antibiotic targeting the 50S ribosomal subunit is, for example, macrolides. These antibiotics generally inhibit peptide bond formation, also preventing protein synthesis. So now we're somewhere else, right? The forming of the peptide bonds, we mentioned that during the P. So it inhibits it here. The other one was blocking the binding site at A. So now it's hopefully beginning to become interesting and relevant to you. Why these details matter? Why are we studying these different subphases? Precisely because we have drugs targeting them differently, leading to different results, etc. And these macrolides, maybe you've heard of erythromycin or azithromycin or clarithromycin. All of these bind to the 50S subunit, preventing translocation of the ribosome along the mRNA. And these macrolides are typically, they are bacteriostatic. So to recap, we had antibiotics targeting the 30S ribosomal subunit. And these typically inhibit translation at early stages. We had aminoglycosides and we had tetracyclines. Then we said we have antibiotics targeting the 50S subunit. And these antibiotics generally inhibit peptide bond formation or ribosomal translocation, also preventing protein synthesis. And here we mentioned macrolides. Excellent. Let's keep going. Now, it's also important to mention genetic variation in bacteria. So we can have mutations, and we have different types of mutations. For example, point mutations, like silent, missense, or nonsense. Here, a single nucleotide change in the DNA sequence that leads to different effects. So if it's a silent mutation, there is no effect on the protein. If there is a missense mutation, it alters one amino acid in the protein, potentially changing its function. If it's a nonsense mutation, it introduces a premature stop codon, leading to a truncated, non-functional protein. And then we have insertions and deletions. So insertion essentially means addition. You're adding something, right? And deletion is removal. So removing something. So it's either an addition, an insertion, or removal, a deletion of nucleotides. And causes of these changes could be spontaneous or induced by mutagens like UV light. So, for example, resistance to rifampin could be caused or is caused by mutations in RNA polymerase. Now, what is horizontal gene transfer? It is the exchange of genetic material between bacteria, occurring independently of cell division. So it is the transfer of genetic material between organisms not involving reproduction. Now, this process plays a crucial role in genetic diversity, adaptation, and the spread of antibiotic resistance among bacterial populations. Now, what is the mechanism? So first we have transformation. It is important to know that it is the uptake of naked DNA from the environment. So bacteria absorb free DNA fragments from their environment and integrate them into their genome. And this requires the recipient bacterium to be competent, meaning capable of DNA uptake. In the Griffith experiment, for example, with Streptococcus pneumonia, the experiment demonstrated transformation where a non-virulent strain acquired capsule genes from a heat-killed virulent strain, making it then pathogenic. So something that was not virulent acquired something, a capsule gene, from something dead, and then became itself pathogenic. So transforming. Then we have transduction. So a bacteriophage, for example, which is a virus. Some people get confused because it contains the word bacteria, but it's bacteriophage. So a bacteriophage, a virus, infects a bacteria, incorporating bacterial DNA into its viral genome and transferring it into another bacterium. So we have two types of transduction worthy of mentioning. One is generalized transduction. So a random bacterial genes are transferred. And then you have specialized transduction, where only specific bacterial genes near prophage insertion sites are transferred. So example, in Corynebacterium diphtheria, it acquires diphtheria toxin genes via bacteriophage infection, and it makes it virulent. Then we have conjugation, which is direct cell-to-cell transfer via pilus. So via the pilus, it's a direct transfer between two cells. And so it requires direct contact between the bacterial cell via this sex pilus. And it involves the transfer of plasmids, if you remember, extra chromosomal DNA, plasmids. Example, our plasmid transfer in E. coli enables the spread of antibiotic resistance genes, making it harder to kill this, to treat this infection. So why is all of this treatment related or clinically relevant? Well, horizontal gene transfer, HGT, is a major driver of multi-drug resistance in bacteria. For example, methicillin-resistant Staphylococcus aureus, MRSA or MRSA, acquires its resistance genes via HGT. Another is virulence factor transmission. Why is it, again, clinically relevant? Because bacteria can gain toxins and other pathogenic traits, increasing their ability to cause disease. Now, genetic elements, transposons. So transposons, or they are also called jumping genes, are these mobile genetic elements that can move from one location to another within the genome. And unlike plasmids, for example, they cannot replicate independently, but they can integrate into chromosomal DNA or plasmids. And an example of a function is that many transposons carry antibiotic resistance genes, allowing bacteria to survive antibiotic treatment. Another thing they're involved in is that some transposons contain genes encoding toxins or virulence factors, which enhances the pathogenic potential of bacteria. So an example of transposon is in vancomycin-resistant enterococcus, also abbreviated VRE. Here, transposons have played a critical role in spreading vancomycin resistance among these species, making it more difficult to treat this infection. So in summary then, bacteria have efficient genetic systems enabling rapid adaptation. Relevant concepts from the lecture included 70S ribosome, and if you remember, for eukaryotic cells we had 80S, and for prokaryotic, like bacteria, we have 70S. And we have the subunits, 30 and 50. And other important concepts, the absence of a nucleus. So eukaryotic cells have a nucleus. Bacteria do not. We mentioned transcription and translation. And it's high yield to remember the initiation, elongation, and termination processes. Antibiotic treatment targets many times specific subunits like tetracyclines target subunit 30S and macrolides 50S. Horizontal gene transfer drives evolution and resistance. And that's the end of the lecture. Thank you for listening. Continue now to Chapter 4.